First of all, thank you for the very much to the organizers and Chairman Dr. Akio. I really appreciate to be here, this kind of exciting meeting. Since this is the international meeting in Japan, I always started with the advertisement in our university, Keio University. Mostly Japanese people know about that. But the important point is, you can see this field. This is the most expensive, money in Japan, 1 million. You can see this guy. This guy established our university 150 years ago. So you remember history of the Keio University. This guy named Junichi Sugahara. So remember. Now, back to the history of not the Keio University, but the history of the gene. Entitled here, System Biology of the Archaeal RNAs and RNA-Binding proteins. Since the jury already talked a lot about the tRNA historical, standpoint, and I think that this is very important finding because of he, already predicted this kind of theory 20 years ago. I just started the tRNA equation study 5 years ago, but we found a lot of unique evidence supporting his finding. We explained this finding to my students. First, but I just started with three dimensions of life. It's the universal origin tree of life. You are here is animals. It's a eukaryote and bacteria and archaea. We are interested in this archaeal stuff. And this is a red region here. Red region, shows that this organism belonging to this area is living in a very hotly, hot circumstances, like 100 degrees around here. It must be related to the original life. Something like this, it's a kind of deep sea vent, water comes out. There's a lot of the organisms collected from here. This mostly belongs to the Archaeal stuff. When you see the inside the Archaeal, I mean the genes. In this case, a comparison with the transcription machinery between the Archaeal stuff and eukaryote. Eukaryote is necessary to start with a lot of the factors to start the correct transcription, like T2B, tata-binding protein, 2B, 2A, or subsequent RNA polymerase. But in the Archaeal, essentially they are similar molecules, or the more poor molecules necessary, like 2 tata-binding protein and T2B molecule, and RNA polymerase, just 3 is enough to start the correct initiation of the transcription. That means that Archaea have a more core component to, compared to the eukaryotic cells. In my talk, I especially use this organism. It's called Pyrococcus pyosus. The pyro means kind of fire, and the coccus means ball, circular orbit is a coccus. That means the fireball is organized like this. You can maybe use in a molecular biology use a PCR enzyme, so-called PFU DNA polymerase. PFU DNA polymerase comes from this organism. One more important thing is that this organism produces the heat-stable protein, like a PCR enzyme. This is very good for the biochemical analysis. And usually the complete genome sequence are available from the database. Plus, the size itself is not so huge, just 2,000 kilobase. This is the half of the E. coli. And number of the open reading fragments is just 2,000. This is small enough to check all of this. That's why we started systematic analysis of this organism. Plus, our aspect is this is a famous figure at the center. Our aspect is focus on the established more than 50 years ago, or something like that. But ribosome from the RNA to DNA, reverse transcriptions appeared. But nowadays is the age of the RNA. RNA regulated the DNA. Also, some of the RNA binds to the metabolite, not protein. So that means that RNA molecules have a network of whole of the central dogma. I'll explain more about this. Why RNA are so important? RNA have a dynamic change of their molecule. RNA splicing and the processing and editing. Compared to DNA, DNA just stock the information of the genetic code. But their case is T cell receptor of the immunogenes. Immunogenes, rearrangement occur during the process of T cell or B cell development. But many tRNA is not tRNA. Many RNA have a splicing machinery. Like one report suggested that more than 90% of the human messenger RNAs have an ordinary splicing or something like that. It's many types of organisms. And after the 2001 or 2002, so many non-coding RNA comes out from the genome sequence. I'm the one of the finder to pick up these non-coding that exist in humans or mice. In the case of mice, maybe 30,000 protein coding genes already annotated. But also we found out more than same class, 30,000 or 5,000, 50,000 non-coding RNA also exist in the genome and expressed. So far, nobody knows the real function of this non-coding RNA. But anyway, it's they exist. So we should consider the definition of the genes. Usually, the definition of the genes is defined as so-called protein coding genes. But so far, we have a lot of the non-coding RNA stuff. We should think about it again. Anyway, so many ribozyme again, and several other stuff. Especially ATP is the energy source, and GTP have some kind of similar transduction molecule, like a GTP binding protein, like transfer some signals. And these two are important substrate for making RNA. It's not DNA, deoxy, but RNA molecule. This stuff is really important. So people just may thought about the RNA world may exist before the DNA world. RNA, in my case, I will talk about the tRNA molecule especially, and also the focus on the RNA-Binding. tRNA is important, already known during Julie explained a lot. But in my point of view, tRNA is a molecule. It's RNA molecule, but involved in the translation. But in some cases, like retrovirus, HIV use the tRNA molecule, RNA primer. This means tRNA molecule used for the DNA synthesis, for example. That means the tRNA involved in every step of the central dogma. That's why when we focus on this molecule, it may be related to the fundamental function of the gene, especially the origin of the genes, genetic code. That's why we developed the new software. Here, we'll explain what we make in the software and how to get the evidence. Actually, we do two ways to make our science. One is a computational science. The other is molecular biological science. He's an expert of the computational science and build up the new software called Sprint. He will explain about our finding using the spread program. See that?
everyone. First of all, I'd like to extend my appreciation to Professor Murase and all staffs of this meeting. I'm very glad to be here today. Now let us begin. My research field is genomic informatics, and I analyze a large number of genomic sequences and predicted the transfer RNA gene sequence. Previously, tRNA scan SE, a program for predicting transfer RNA genes from the genome sequence, has developed. This program detected the clover-leaf structure, a well-known structure and a well-known and unique feature of transfer RNA molecule. And tRNA scan recognized this region as tRNA gene candidate. Also, this program can predict normal types of tRNA. This program can predict normal types of tRNA. It sometimes mispredict unusual types of tRNA, such as intron-containing tRNA, mostly found in Archaeal genomes. It is because that intron in the tRNA inhibits the formation of clover-leaf structure, and tRNA scan C cannot recognize this structure as a tRNA. Therefore, we developed the SPRITS, a new program for predicting intron-containing tRNA. Our program is constructed by very simple algorithms. First of all, SPRITS predict the intron sequence, not tRNA sequence, just predict intron sequence, and then removes it and join the exon sequence, and the intron-free sequence is created. Then this intron-free sequence is queried to the tRNA scan SE. This simple step enables tRNA scan SE to detect intron-containing tRNA as a normal tRNA. To date, we analyzed more than 50 Archaeal genomes, and we detected total 2,400 tRNA genes, including almost all missing tRNA in Archaeal, and that cover more than 99.9% of codon tables in Archaeal. In this process, we founded some novel types of tRNA. For example, this is multiple intron containing tRNA that possess 2 or even 3 introns at single gene. We consider that the presence of this tRNA suggests the dynamic and complex splicing regulation might be present in the archaeal cell. Moreover, using SPRITS, we also founded the first case of the permitted tRNA gene in which 3' half of tRNA lies upstream of the 5' half in the genomes. In other words, for example, this interesting tRNA is created by the permutation of the genomic information. First of all, 5' and 3' ends of the precursor tRNA is joined to create the circular RNA intermediate after the transcription, Then, opposite side is processed to generate the correct termine. So, through this process, first information on the genomic region, in this case Tokyo, is permuted to Kyoto. Moreover, we founded the tri-split tRNA, which is made from 3 individual small RNAs that transcribe from 3 different genes. This is very interesting tRNA and novel types of tRNA. And about tri-split, descriptions will be explained by Professor Kanai. So my turn is finished. Thank you for listening.
Thank you, Darwin. Okay, he developed the split tRNA database also. The split database is available on the internet. We reported the result of our database in last year, molecular biology and evolution. Anyway, this software is very unique and 99.9% just, oops. 99.9% of the tRNA are completed, but one organism called hyperthermic acidic Archaeal, cultivar. This is a unique Archaeal. It's living in the Philippines, the hot spring. But we have a difficulty even in his program, to predict these very 6 genes and find out these genes, they are first case of the split gene in the free living organism. Since the nano-Archaeal is some kind of reduced genome and symbiosis of the certain Archaeals. That's why this is the first case of the free living organism having the split DNA. I explained the first example, the alanine group here. These 3 split genes is split into 2 pieces together. This is kind of the tag region. And after the tag region hybridized, it's spliced out to this region and makes it mature here. And tri-split return is made from the 5 different transcripts. Namely the 1, 2, 3, 4, 5. This 2 is the most important fragment. It's a kind of a template fragment. It's 2 hybridized with the 1, so makes it a glycine TCC. Not this one. CCC. This just comes from 2 fragments. But in some cases, this one is separately encoded to the 2 different fragments, 3 and 4. This makes the TCC, and some is just like this, cassette changed from the 5 transcript. This is the GCC right here, and this, 5 choose of the 5 transcript make the 3 types of the tRNA. This is a tri-split, and this is a split tRNA. This is the first case of the, just like jigsaw puzzles, make the tRNA mature molecule from the fragments genes. And another example is, since this is a really rare tRNA, we just left it examine this is really, amino-translated or whatnot. We isolated the glycine amino acid tRNA synthase from this organism and make up the amino acylation assay. And you see there, in this case, this is a tri-split, and this is a split tRNA. The both cases in a kind of a mixture of the glycine RS plus glycine. You can see the triplet, but this indicates this actually amino acylated and can work for ready for the protein synthesis. Plus, one more thing you see there. When you compare to this hybridized region to neighborhood of the intron sequence, first of the when you compare to this sequence, this is the split tRNA tag sequence, so-called linker sequence. It's really a high homology with the neighborhood with the intron sequence. If this are in there, something like this, but actually the additional sequence some here in the case of the split tRNA, but you can see that lot of the conservation like here. So we can imagine there must be an evolutionary relations between the split tRNA and intron-containing tRNA. So we don't know which is first, but from the the concept of the jury, split gene must be appeared first because it's really difficult to understand if mature tRNA gene into the split two in the later stage, because if this is true, we have another promoters here or something like that. It's radius how it is seen. So this might be a really primitive stage of the tRNA, because we just find out that this primitive tRNA is a really unique type of the organism, especially living in a hot spring and related very rare stage of early stage of their life. And because of the sequence of the tRNA sequence available based on the Sugahara database, we additionally analyze the sequence of the Archaeal genome of the tRNA through the combination of the ancestral gene as a 5' and 3'. This is so-called the network analysis. We firstly collected the whole mature tRNA sequence and divided the 5' half and 3' half and make the network analysis. Network analysis means this is a kind of a threshold of the homology of the genes. When the threshold is 7%, its tRNA 5' half is just like almost one sequence. But when you are increasing the specificity, the tRNA is divided into its own sequence. Like this must be some kind of setting sequence or something like this, but not all. But when you compare to the 5' half, 3' half more, high stringency, but a high threshold necessary to separate each of their fragments into their corresponding to each amino acid. That means if the 5' half and 3' half is the same background, evolution, this must be a similar structure of the network. But this result suggested from the sequence of the 5' half and 3' half is different genetic background. That's why we collected these categories to make up the codon table fields. Successfully we can see that some codon table is most of the older codon table can fill up with a mix of this kind of mixture. Also, we have collected several examples there. In this case, 3' have to really conserve maybe 95% of the homology. But in this case, it is different. This is the non-Archaeal isofusin and glycine. The 5' up is different. In this case, the 3' up is different. So it may be possible to have some kind of a combination of the 5' and 3' up makes the mature here. Now we need a more example. Since our institute located very northward in Japan, we have a lot of the hot spring. We collected the environmental sample from the hot spring. This Japanese data about the hot spring. Actually, this hot spring is very near, to in our institute. We collected from the depths of the 1 kilometer and temperature about 60 degrees. He's one of my students, undergraduate student. He collected this, hot water and centrifuges, and we just got the small amount of the, microflow. We make the cDNA library of this hot spring. Actually, we got using the next generation sequence, and we already got the 80,000 tRNA sequence from the environmental sample. Because we only know maybe some people calculated 1% of the microlog sequence available. We don't know the 99% of the microlog. We only use the lab strain of the like E. coli, bacillus. This is just the part of our knowledge of the genome sequence or the cDNA sequence of all micrologs. That's why we need to collect more samples from the environment. This is the ongoing project. Okay. Lastly, I just talk about the more focus on the RNA-binding protein, not only the tRNA, but RNA-binding protein also very important because RNA and protein may coevolve. Like here I asked about the coevolution of the tRNA molecule and some enzymatic, stuff. This is also to, the ribosome RNA also bind to the ribosome protein. tRNA bind to the tRNA amino acid system. So coevolution is important. That's why I started with the, systematic analysis of the biological enzyme. Because this bacteria I told you, it's just produced 2,000 proteins. And I just focus on the molecular biological way of our proteins. And we just predicted more. One of the problems is after the genome sequence, the complete genome sequence, half of the protein belong to the unknown function, but just conserve hypothetical function. This is a really problem. And this is because, mostly the annotation of the protein is based on the alignment of the known sequence. That's why, homology sequence analysis to determine the function of the proteins, I don't like that. I really like to determine the protein function based on their function. This is a functional screening and feedback. This is a strategy of our cloning methodology, the expression cloning of the novel RNA-binding proteins. We firstly got the genomic DNA from the Pyroglobulinous organism and cut it into small pieces and collected 7 kilobase DNA fragment and ligate to the plasmid. We prepared the whole numbering of the plasmid DNA and makes the plasmid file. We numbered from 1 to 1,000 independently. And each plasmid clone transform the E. coli, another E. coli. And E. coli expressed the exogenous phytococcus proteins. The important thing is that after the expression in the E. coli, we heat treated the E. coli at 85 degrees for 15 minutes. This is a good for kill the endogenous protein from the E. coli, but not the phytococcus protein. So after the centrifugation, the supernatant that each tube have a recombinant phytococcus protein, it's produced heat stability. So we have the protein pools numbering from 1 to 1,000 and the plasmid pools. If I have some kind of activity from the tube, we are automatically back to the plasmid data. That means if I find some function here, we know the genes. Since they are just genome size is 2,000 kilobase, we have a 7 kilo fragment, 7,000 kilobase is enough to whole genomes. In this way, I found a lot of the RNA-binding protein. One example like this, for screening, we use the zero-shift analysis. The probe is something like this structure. This happened to, there are some AUG sequence in the loop region, the secondary structure. This structure is very unique for the 5 upstream of the certain messenger. In this case, the Saigon messenger. I don't know the exactly of the Saigon gene so far, but in the first time. But when I clone the genes corresponding to the RNA binding of this particular sequence, it's turned out it's a thymidine messenger. Because this product is a thymidine synthetaser, this is very important because thymidine is making T of the DNA. T is really specific for the DNA, not U. A, T, G, C, and T is specific for the DNA. So what we found is T molecule synthesis is RNA under the control of the RNA. So it's some kind of relationship there, thymidine synthesis is under the control of the RNA regulation. That's why I like this example. Another example is we found out some kind of a ligase to ligate this particular position after the removing the intron. It's already reported some terminal structure is important. But this should be some cyclic form of phosphate and this OH. I tested several terminal structure, and we found out from the library, PF27 protein have ligation activity of the tRNA hubs. It's already reported that yeast and plant have a tRNA ligase, but nobody knows in the Archaeal and human the tRNA ligase. But this is a very unique stuff. But this also have some kind of problem. I explained it. Anyway, we successfully ligated the tRNA hub in vitro. And this is required some GTP. This is also the unique characteristic of the tRNA ligation. But the problem is after the several experiments, we found out that this ligation occurred at 2' and 5'. This is a very unusual structure of the nucleic acid. Should be the 3' or 5'. But anyway, this is the first example. We just studied the going on. This is you know the most of the case, but one enzyme in the yeast and the plant can do this, but no homologs of the tRNA ligase of the yeast or plant in the human Archaeal. That means tRNA ligase yes, but our candidate is the ligase tRNA 2' and 5'. This is a stretch. After these circumstances, there are some nature papers said that human RNA kinase called human FIP1 works on both the tRNA ligation and some phosphorylation of the siRNA. siRNA is the famous RNA to the RNA interference. But anyway, this enzyme is necessary for the tRNA ligation also. In the RNA meeting, I talked with these two guys, and it must be a human ligation related to the Archaeal ligation. So I found it, actually, human siRNA kinase homolog in the Archaeal from Biosciences, and checked that this is actually occurred using ATP and magnesium and the phosphorylation occurred. So tRNA ligation is a human mechanism is human archaeal DDT data. But interesting is, this archaeal tRNA, not tRNAs, archaeal phosphorylation enzyme phosphorated DNA as a substrate. Human enzyme not like this, it only likes the RNA. But the archaeal, phosphorylation enzyme likes the DNA also. They even can use the deoxy-DDT and deoxy-ATP as a substrate. So so far we constructed some model of the 3' 5' ligation, but center position is not, we don't know it so far. Another report in the last year, Dr. Ho's group in Buffalo, USA, they identified a new type of RNA ligase. This is our enzyme. This is really necessary to the tRNA ligase, but it's a problem with the 2' 5' ligation. But they are finding there T4 types of RNA ligase. It's not terminal tRNA splicing, but this can ligate there, very effectively. But interestingly, this enzyme also ligate RNA and DNA. This suggests that the archaeal enzyme vaguely recognize DNA and RNA. It's not strict. The final example is here. Our study is here. Life looks like the same, but the different point is that amino acid alignment is the first eventual structure of amino acid DDT. So we compare the amino acid sequence. The sequence is different, but some partial structure looks like the same. In this case, the secondary structure. We mapped the evolutionary conserved amino acid among the RNase H or the E side among the other proteins, and find out that this region is both helix, but also indicated that this particular region is all, having the amino acid. In this case, conserved RNase, but in this case, conserved other protein. So we make the kind of the chimeric proteins with this particular region. We just focus on M-nana example, just one amino acid replacement. And this amino acid replacement is enough for decrease the activity for unexpected activity. Usually, RNase H is digested RNA, DNA, some like it. This is a both enzyme. This is the wild type. This is the purification table of their SD space. This is the ion exchange chromatography. The peak is 910. 910 peak is produce the degradation activity, the substrate, the RNA, DNA. But interestingly, this archaeal enzyme also copies the RNA, effectively. But the one point mutation is decreased RNA, activity, but not DNA, RNA activity. We examined this position with another example. M6, M9 also the similar activity, decreased activity. We mapped the model of the RNase enzyme with the substrate. Usually this red region is necessary for cutting the RNA molecule from here. But we found amino acid is here, right here. Right here, just only touch to the template, not digestive structure, but the template structure. We suspected that this region may recognize this is in RNA or this is the DNA or something like that. Anyway, after this, we may also vaguely recognize DNA, RNA. From this result, this is a conclusion. Evolutionary biological system is coming from the simple to the complex. Like a tRNA molecule is a simple fragment to the first and makes the complex system in a later stage. And the archaeal enzyme still have a value non-specifically to the recognition of the DNA RNA and makes specific recognition to the